Hey folks, Professor Finn here, and I'd like to welcome to you to this lecture series on microbiology. Now, the textbook we're going to be using here is Pathology and Microbiology for Funeral Science Students by Mullins. There's an older version, which is a purple cover. There's a newer version, which is a tan cover with a red insert. Either of those books will be generally fine. This lecture was created using the older version, the purple cover, then I updated it when the new the new book came out. There are some changes between the books. The appendices, I believe, were removed from the later edition. And I will refer to one of those appendices during this lecture series. So, like, share, subscribe, let people know I'm out there and I exist. Share this with a fellow Mortuary Science student friend or a person who is interested in the subject. Let's go ahead and get the screen shared, and let's talk about the introduction to microbiology. Let's get the pen up and running, and away we go. Before the 19th century, it was commonly believed that life forms could spontaneously appear from non-living matter. This spontaneous generation was believed to occur most frequently from dead and decaying matter. Your book references a plan for generating mice using sweaty underwear and husks of wheat to be placed in an open-mouthed jar. And let's be grateful science has improved since then. In 1668, Francesco Redi challenged the theory of spontaneous generation. He believed that maggots arose from the eggs left on meat by flies. He ran an experiment and found this to be the truth. This did nothing to change everyone's outlook on things, and even Redi himself believed that spontaneous generation could occur under certain circumstances. Surprisingly, even the development of the microscope initially strengthened the theory of spontaneous generation. This was a result of putting hay into water and waiting for the animalcules to appear. In 1745, John Needham, an English clergy, tested the theory of spontaneous generation. He used chicken broth, which he poured into flasks and then sealed them. The broth became cloudy and, hallelujah, praise Jesus, spontaneous generation won. A gentleman by the name of Lazzaro Spallanzani once upped the experiment because he believed that the broth became contaminated by the air as it was being poured into the flask. He replicated the experiment, induced a vacuum, and then boiled the broth. No organisms appeared, but scientists of the day said that was a result of the lack of air being necessary for spontaneous generation. So always remember, when people say trust the science couldn't agree with the science. It was not until 1859 that the theory of spontaneous generation was dispelled and scientists began to recognize that spontaneous generation just wasn't possible. Enter Louis Pasteur. Pasteur boiled broths in a flask and then heated the glass neck of the flask until he could bend it into an S shape. This allowed the air to reach the broth, but airborne microorganisms settled into the curved neck of the flask due to gravity. After no organisms grew in the broth, he tipped the flasks, allowing the broth to reach the curved neck of the flask. Now faced with a free lunch, the microorganisms multiplied, and it was proved the theory of spontaneous generation was crap. It also proved that microorganisms are everywhere, even in the air. Rudolf Virchow, or Virchow. The modern medical community accepts Rudolf Virchow's theory of biogenesis, which he put forth in 1858. The theory of biogenesis is based on the principle that no life form can spontaneously appear in living cells and can only arise from pre-existing living cells. It is therefore possible to control the spread of microorganisms and impact conditions of disease. Modern aseptic techniques are the standard practice in laboratories and medical procedures, including embalming. Vaccination. It has only been since the early 1900s that medical science has recognized that microorganisms cause disease. Before then, germ theory was not accepted by the scientific community, and most effective treatments for disease were simply a matter of getting it right, good old-fashioned trial and error, with no actual understanding of what is actually happening. Again, makes you proud. Enter Edward Jenner. Edward Jenner trained in London as an army surgeon before returning to his native Gloucestershire in the west of England, where he spent his entire career as a country doctor. In 1798, Jenner tried to find a way to protect people from smallpox. Building on a previously developed method of vaccination against smallpox used in Turkey and his own observation that milkmaids who developed cowpox were immune to smallpox, he inoculated healthy volunteers with material scraped from cowpox blisters. This sounds like fun. After a mild illness due to inoculation, the volunteers did not develop cowpox again and they did not contract smallpox. The process became known as vaccination from the word vaca for cow. Germ theory of disease. At one time, it was common religious belief that disease was a punishment for crimes and sin. You're going to hell! In 1665, Robert Hooke reported that the smallest structural forms of life were little boxes or cells. His observations formed the basis of cell theory. Cell theory contains that all living beings are composed of individual cells. Between 1674 and 1683, Anton von Leeuwenhoek reported the observation of microorganisms or animalcules. He observed them in rainwater, like lake water, on the surface of teeth, in the gut of horseflies, and in human diarrhea. Hope you wore gloves. The theory of spontaneous generation was not dismissed overnight by the theory of biogenesis, and likewise, the acceptance of germ theory was not generally established until 1900. In 1835, Agostino Bossi proved that a particular type of silkworm disease was caused by a fungus. In the 1840s, Ignaz Semmelweis demonstrated that childbirth fever could be spread among patients by physicians who did not disinfect their hands. In 1865, Louis Pasteur found that a protozoan caused a silkworm disease that threatened the silk industry throughout Europe. Also in the 1860s, Joseph Lister, more on him later, applied the germ theory of disease to the treatment of his patients during medical procedures. Although disinfectants were unknown at the time, Lister recognized that carbolic acid, or phenol, kills microorganisms, so he began treating surgical wounds with a solution of it. Infection as a result of surgery was a common cause of death, and even now, phenol is a common component of many chemicals used in the modern embalming facility for this reason. In 1876, Robert Koch established the causation of disease by microorganisms. As anthrax killed numerous cattle and sheep throughout Europe, Koch and other scientists were attempting to discover a method of containing the spread of the disease. He discovered the existence of rod-shaped bacteria, Bacillus anthracis, in the blood of cattle that had died of the disease. After playing mad scientist and culturing the bacteria on nutrients, he then injected them into healthy animals. After comparing the bacteria found in the healthy animals with the bacteria of the animals that originally died of the disease, Robert observed that the bacteria were the same within the two sets of blood cultures, and it still sucked to be the cow no matter what. For the past century, scientists have used a sequence of steps to determine the specific microorganisms that cause a specific disease based on Robert Koch's original steps. There are 4 steps to Koch's postulations. First, the microorganism must be present in every case of the disease. The microorganism must be isolated from the host with the disease and grown in a pure culture. The specific disease must be reproduced when a pure culture of the microorganism is inoculated into a healthy, susceptible host. The microorganism must be recoverable from the experimentally infected host. Once germ theory became the hottest thing since radioactive fidget spinners, scientists agree that this effect could control microorganisms outside the body. This led to one idea that a chemical known as the magic bullet could be used to destroy disease-causing microorganisms in the body without harming the infected person, and that's how we launched the pharmaceutical industrial complex. Paul Ehrlich was a German physician who studied the staining of animal tissues as a medical student. The theories and methods he used, described in his dissertation, is the basis of many of the staining techniques in use today in microbiology labs around the world. In 1899, he began researching for a magic bullet that would treat syphilis. Your book has a list of common diseases, the microorganisms that cause them, and the pharmaceutical or treatment that kills them. know it. So looking at you, table 20-1, these charts are the absolute best way for you to study these things as you progress going forward. Grab a chart, look through it, and do your thing, okay? People might not always have the time to read the chapters, but you have time to look at a chart. You can just flip right through it, okay? And here's a hint. Penicillin, tetracycline, that kills damn near anything, okay? What you're focusing on are the ones that stand out as the only thing that may affect a certain type of disease. Those are the ones that get special attention. You may ask how much of this chart you may actually have to know. I promise you, the disease and the bacteria are really, really important. You may also know that penicillin is basically the Ubermensch of killing most things. Closest thing to a magic bullet that we have. By 1907, Ehrlich and his assistants had tried hundreds of chemicals in an attempt to treat syphilis. They discovered the usefulness of arsenic-based drugs against protozoan infections that caused African sleeping sickness. Close, no cigar. You might have killed African sleeping sickness, but syphilis still for the win. The 606th drug in the series had already been rejected as a treatment for syphilis. However, Ehrlich and his assistant tested this arsenic-based drug again on rabbits that they infected with the spirochete that causes syphilis, Treponema pallidum. Ehrlich also found that the 904th drug tested also effectively treated syphilis. He then tested both of these drugs together hundreds of times. He reported that both salvarsan and neosarvarsan were effective in treating syphilis. By the late 1930s, researchers had determined that many synthetic drugs were effective agents in the treatment of infectious diseases. Many of these were derivative of dyes based on Ehrlich's early studies as a medical student. Despite all of this, there is no magic bullet that can treat all disease. All medications have side effects on the body, and the mixing of medications with other prescription medications can lead to complications and death. Even holistic medicine, vitamins and mineral supplements, and non-prescription medication can interact with prescription medications and cause complications. This is why it is vitally necessary that when you go to your medical professionals, you be very honest and truthful with what you are taking, what you are eating, what type of vitamins you're taking, so that you don't have any nasty interactions. A big concern to us as funeral professionals is the effect of these medications on the condition of the remains and the interaction that some of these pharmaceutical agents may have on the embalming process by messing with embalming chemicals. It's also helpful to be aware of radioactive chemicals that are used in the treatment of cancer. Now, I have an entire lecture on how these things interact with the embalming process, and the long story short is the medicines themselves we don't typically care about. What we care about is the effect, the side effects that the medications caused in the body, the renal failure, the liver failure, the ulcerations, etc. Those are things that matter to us in the preservation equation. Not all medications are synthetic. The first antibiotic was discovered by accident in 1928 by Alexander Fleming. This genius let his culture plates be contaminated by mold. Let that sink in. Wasn't looking for it. He done effed up, and he still won. Unlike the arsenic or sulfur-based drugs that had specifically been studied for their affecting its microorganisms, the natural ability of this fungus to inhibit the growth of bacterial cells was unexpected. The specific mold was later named penicillin, and it was originally produced on corn and decomposing cantaloupes in Illinois. Before World War II, approximately 400 million units of penicillin had been produced, and by the end of the war, U.S. companies were producing 650 billion units each month. Think about how many that is. Just go ahead and multiply that with a profit of 1 penny per unit, and then multiply that by 12. Ridiculous. Naming microorganisms. Microbiology is defined as the study of microorganisms and their effect on other living organisms. Micro is a prefix that means extremely small and the other word bio means life. So microbiology literally means the study of extremely small life. Your book references zits and little critters that contribute to its formation, which is Staphylococcus aureus. We'll talk about what Staphylococcus, all that means, another chapter. The Latin names of the bacteria will give you a valuable clue as how to name them. So, for instance, the word coccus means shaped like a sphere or a ball. Staphyle means clustered together, similar to grapes. Aureus means a golden yellow color. So, this microorganism literally means it is a cluster of little spheres that are gold. That's what it does. In 1673, our buddy Anton van Leeuwenhoek saw red blood cells for the first time through microscope, and he was in the same position as the little green people your book was just talking about. Early scientists like him wanted to differentiate one microorganism from another, so they began to describe them in even smaller groups by their characteristics. The same basic groups are still in use today and form the backbone of an understanding of all life forms. Eukaryotic versus prokaryotic. Every living organism has cellular properties, and these properties are based into two broad categories. Eukaryotic cells and prokaryotic cells. The word eukaryote means true nucleus. Eukaryotes are usually distinguished from other forms of life by the presence of nuclei and a cytoskeleton. Most human cells are eukaryotic. Their cytoskeleton comprises a variety of proteins. The cytoskeleton provides shape for the cell and support for membrane-bound organelles, and the nuclei contain genetic information organized into discrete chromosomes and contained within a membrane-bounded compartment. The most evident organelle in most cells is the nucleus, and it is from the presence of this organelle that the eukaryote gets their name. Most cells have a single nucleus, while some have thousands, and others, like red blood cells, have none, but can be shown to derive from cells with nuclei. The nucleus is bounded by a membrane called a nuclear envelope. Speaking of the nuclear envelope, the envelope is perforated by nuclear pores that allow compounds to pass between the nucleus and the surrounding cytoplasm. Some eukaryotes, called protists, have more than one kind of nucleus. One contains copies of reproductive material, and the other regulates activities within the cell. The big difference between prokaryotes and eukaryotes is that eukaryotes have a nucleus and a membrane-bound organelles, while prokaryotes do not. The word prokaryote means before nucleus. Cell differences. The DNA of prokaryotes floats freely around the cell, whereas the DNA of eukaryotes is held within the nucleus. The organelles of eukaryotes allow them to exhibit much higher levels of intracellular division of labor than is possible in prokaryotic cells. Eukaryotic cells are, on average, 10 times the size of prokaryotic cells. Eukaryotes are larger. Okay? Eukaryotes are larger. The DNA of eukaryotes is more complex and extensive than those of prokaryotes. Prokaryotes have a cell wall composed of peptidoglycan, which is a single large polymer of amino acids and sugar. Many types of eukaryotic cells also have cell walls, but none are made of peptidoglycan. The cells of animals, plants, protozoa, and most algae are typically eukaryotic. The cells of bacteria and cyanobacteria, or blue-green algae, are prokaryotic. Kingdoms. There are 5 kingdoms used to classify all biological forms, which include all of the eukaryotes and the prokaryotes. The 5 kingdoms are Monera, Protista, Fungi, Plantae, and Animalia. Okay, Monera, also known as Prokaryote, Protista, Fungi, Plantae, and Animalia. These 5 kingdoms classify all life forms by their shared characteristics and are divided into successively smaller groups. Each group classifies a particular life form from a larger group. The order of classification, known as the taxonomic rank, going from largest to smallest, is kingdom, division, class, order, family, genus, and species. So kingdom, division, also known as the phylum, class, order, family, genus, species. The genus and species are the most important for the naming of microorganisms. Monera. Monera is the only kingdom composed of prokaryotic organisms which have a cell wall and lack both membrane-bound organelles and multicellular forms. Protista. This kingdom includes a variety of eukaryotic forms. Protists are best defined as eukaryotes that are not fungi, animalia, or plants. Okay? So, protists are eukaryotes, but they aren't funguses, they aren't animals, and they're not plants. Fungi are eukaryotic, multicellular groups of cells with multiple nuclei enclosed within cell walls. Plants are immobile, multicellular eukaryotes that produce their food by photosynthesis and have cells encased in cellulose cell walls. And animals are multicellular eukaryotes capable of mobility at some stage during their lives, and their cells lack cell walls. Microorganisms have a first and last name just like people. Your book notes that Western cultures have a given name first and a surname last, with each being capitalized, while some Eastern cultures do the last name first and the first name last. And no one cares. Microorganisms are named by two names as well. The first name refers to the genus, and the second name refers to its species. The genus is always capitalized, and the species is always lowercase. Hence, the proper way to spell everyone's favorite staph infection is Staphylococcus aureus. The names are written in italics out of custom because it's not English. Once names are written fully, is it acceptable to abbreviate them? The species name is not abbreviated, only abbreviate the genus. And the reason for this is reference, okay? Because if you have an S, you don't know if that means Streptococcus, Streptobacillus. You have no idea. So you have to say it once in order for people to know what you're talking about. Then after that, you don't have to type the stupid word out again and again and again and again, because it's annoying. Divisions of microbiology. There are 5 important divisions of microbiology. Bacteriology is the study of bacteria. Rickettsiology is the study of rickettsias. Virology is the study of viruses. Protozoology is the study of protozoa, and mycology is the study of the study of fungus. Pathogenic microorganisms. Not all microorganisms cause disease. We'll talk about those relationships in an upcoming chapter. Beneficial microorganisms are used to make food, in treating infections, in water treatment facilities, and in the human digestive tract. With that being said, there are also many microorganisms whose sole purpose in life is to make your life awful. Microorganisms that cause disease are known as pathogenic microorganisms or pathogens. The big jerks that are major health importance to humans are bacteria, mycoplasma, rickettsia, chlamydias, viruses, protozoa, fungi, and prions. The big ones that we will study, each getting their own lecture: bacteria, mycoplasmas, rickettsias, chlamydias, viruses, protozoas, funguses, and our friend the prion. First up, bacteria. Bacteria are defined as prokaryotic, one-celled microorganisms of the kingdom Monera, existing as free-living organisms or as parasites, multiplying by binary fission and having a large range of biochemical properties. This is the definition of bacteria. Period. End of story. Prokaryotes belonging to kingdom Monera. They're free-living organisms or parasites. They multiply by binary fission and have a large range of biochemical properties. The majority of infectious diseases that we will look at are caused by bacteria. That chapter is awful. They are small and must be viewed individually with a microscope, but they are about 10 times as large as a virus. When they are grown in a proper nutrient source, they form colonies containing billions of bacterial cells that can be seen without a microscope. Ricketsias are a genus of rod-shaped, gram-negative intracellular parasitic bacteria. They can also come as a sphere-shaped or can change shape. Rickettsia can only reproduce within a host cell, which makes them similar to viruses. They are extremely small and usually spread by arthropod vectors such as lice, fleas, ticks, or mites. Except for Rickettsia prowazekii, our buddy who causes endemic typhus, Rickettsia do not contain flagella. Rickettsia must have living cells to replicate and are obligate parasites, meaning they cannot survive outside of the host cell. Rickettsia are dependent on the rich cytoplasm of host cells due to the bacteria's uniquely permeable and unstable cell membrane. Rickettsia typically infect the epithelial lining of the intestinal tract of the arthropod vector and are excreted through the vector's feces. These bacteria are commonly transmitted from the arthropod vector to humans through bites. In infected humans, rickettsia are commonly found in the small blood vessels of the brain, heart, and skin. The end result is a blood clot that can lead to the loss of plasma, decreased blood volume, and fatal shock. Rickettsia for the win. It is speculated that rickettsia produce a toxin as well. Rickettsial infections are commonly treated with drugs like tetracycline. Sulfonamides can actually increase their growth, which means you don't want to treat them with it. Chlamydia are a group of non-motile gram-negative intracellular parasites that can cause disease in humans. There's the definition of it. Although chlamydial diseases manifest in a similar manner to viral diseases, chlamydia are not viruses. They are bacteria that replicate in the cytoplasm of host cells. The spherically shaped chlamydial cell contains no flagella or capsule. It does contain a rigid cell wall and often a cell membrane consisting of a large quantity of lipids. Chlamydia are believed to be dependent on host cells because the bacteria lack a source of energy production and require the adenosine triphosphate, or ATP, found in the cytoplasm of the host cell. In humans, chlamydia can cause infections of the eye and genital urinary infections by infecting the mucous membranes in those areas. There are also 100 species of birds that naturally carry chlamydia. If the chlamydia from the birds are passed to humans, they can affect the mucous lining of the respiratory tract. In addition, chlamydia can cause infections of the liver, kidney, and heart of humans, as well as spontaneous abortion in pregnant women. Protozoa. Protozoa are of the kingdom Protista and a one-celled eukaryote, although some may be colonial with various mechanisms of motility. Protozoa lack a cell wall, but may have rigid forms that change based on their development during their life cycles. Although the majority of protozoa are free-living organisms with no disease-causing potential in humans, certain protozoa do cause serious diseases. There are more protozoa in the world than any other microorganism. You know that is hugely important. There are more protozoa in the world than any other microorganism, and they play an important role in consuming both bacteria and multicellular organisms. There are 4 classifications of protozoa: flagellates, amoebae, sporozoans, and ciliates. Protozoa are responsible for diseases such as amoebic dysentery, malaria, toxoplasmosis, African sleeping disease, and a bunch of other fun stuff. Fungi are a group of often filamentous unicellular and multicellular organisms lacking chlorophyll and usually bearing spores. Yeasts, mold, and dimorphic fungi are the 3 fundamental categories of fungi. A fungi that typically has a capsule and is unicellular is known as a yeast, while molds are filamentous and multicellular. Dimorphic fungi alternate between one and the other. Since fungi lack chlorophyll, they often grow on decomposing matter and are called saprophytes. Saprophytes require dead or decaying tissue. Fungi may produce either sexually or asexually through budding or reproductive spore formations, and some fungi produce toxins that are harmful to humans. Yeast can be used to make bread, or they can be used to make you sick. Molds are essential to produce cheese and penicillin, but aspergillus can be toxic to humans. Almost all the world's commercial penicillin is produced from the fungus Penicillium chrysogenum. Human fungal diseases are characterized by the level of tissue penetration. Superficial mycoses penetrate the least and are found only in the outermost layers of the skin and hair. Cutaneous mycoses are fungal infections of the skin, hair, and fingernails such as ringworm, jock itch, and athlete's foot. They are caused by dermatophytes and they occur deeper in the integument than the superficial mycoses. Subcutaneous mycoses are often the result of trauma to the integument and they frequently require surgical removal. Systemic mycoses spread throughout the body and generally originate in the tissues of the lungs. Persons with suppressed or compromised immune systems are more susceptible to the opportunistic fungal diseases, which can result as a secondary infection due to treatment for their original disorder. The elderly, children, and persons with AIDS, persons with diabetes, and persons receiving chemotherapy for cancer could all be described as having suppressed immune systems. The one thing that you're going to see time and time and time again is who's at risk? Children, the elderly, and people with compromised immune systems. End of story. Those are the three people that are mostly at risk. Almost 100% of the time, that's the answer. Fungal infections can be fatal and are often extremely difficult to manage. They can also be passed from the deceased to the living by a variety of means. Deep breath. Viruses, viroids, and prions. The smallest category of disease-causing microorganisms is the virus. A virus is an intracellular infectious parasite capable of living and replicating only in living cells. Viruses range between 200 and 300 nanometers in size. A nanometer is 1/1000th of a meter. There are approximately 25 million nanometers in an inch, and approximately 84,000 of the largest viruses could be lined up side by side in an inch. Viruses come in a variety of shapes and sizes. Your book references a more complex model known as a bacteriophage that has a picture of it in figure 20-5. Again, these numbers are based off of the purple edition of the book and may be changed in the most current version. Viruses can affect almost any living cell, including bacteria, fungi, parasites, plants, animals, and human cells. They're basically a piece of genetic material wrapped in protein, and they contain only one type of nucleic acid. They're either DNA or they are RNA, no ifs, ands, or buts. They multiply inside living cells by using the cell's own structures to produce more infected cells. There are many ways to classify viruses, and no single way has proven satisfactory. So here is an example chart from the book that shows some common virus groups and the example of the diseases they cause: pox virus, herpes virus, adenovirus, rhabdovirus, arbovirus, etc. Viruses lack enzymes like adenosine triphosphate for producing energy, so they are dependent upon the host cells. Some viruses are sensitive to disinfectants due to the presence of lipids in their coverings. HIV can be inactivated by a solution of 10% household bleach in water. Generally speaking, folks, if you need it dead, bleach will do it for you. Bleach might destroy everything around it, but bleach will kill it. Both detergents and ethers are capable of inactivating many viruses by simply dissolving their lipid coverings. This is not unlike the ability of dish soap to dissolve fat from a dirty skillet. And you know the author is not from the South if they're throwing fat away from a skillet. I mean, come on, man. Bacon grease. Viroids. A viroid is similar to a virus, except it only contains RNA and does not have a protein coat. They are intracellular parasites that cannot survive outside the host cell. Both viruses and viroids create an immune reaction response in humans. Hepatitis D, as in David, is caused by a viroid. It is actually contained in the capsule of the hepatitis B virus. Your book notes that funeral practitioners are at increased risk to blood-to-blood exposure, and a forensic pathologist once contracted hepatitis B when he scraped his knuckles against the exposed ribs during an autopsy. Which, when they go after them with the rib cutters, they create some sharp edges. Just letting you know. Prions are small proteinaceous infectious particles that are resistant to most procedures that modify nucleic acids. Unlike their other bodies in this category, prions do not contain either RNA or DNA, and they do not cause an immune system response in humans. Super, super, super important. Viruses and viroids produce an immune response. Prions do not. Prions consist entirely of proteins that are produced by human genes and are obligate intracellular parasites. Viruses, viroids, and prions are all non-living agents, hence they can only multiply inside living cells. Diseases caused by prions are collectively known as spongiform encephalopathies because they leave holes in the brain tissue that resemble a sponge. Prion-related diseases may either be inherited or develop sporadically and can be transmitted through blood-to-blood contact. The common denominator among all prion disorders is the presence of amyloid deposits in the tissues. Amyloid is a general term for any intracellular or intercellular starch-like protein deposit. These deposits stain easily with Congo red dye and appear homogeneous, waxy, and translucent. Amyloid degeneration also occurs in non-prion-related pathological disorders like tuberculosis, osteomyelitis, leprosy, Hodgkin's disease, and various cancers. Prion diseases such as scrapie and mad cow disease, or bovine spongiform encephalopathy, are found in sheep and cows respectively. Prions are suspected causes of human disease such as Creutzfeldt-Jakob disease, also known as Creutzfeldt-Jakob disease, variant Creutzfeldt-Jakob disease, Kuru, Gerstmann-Straussler syndrome, Alzheimer's disease, fatal familial insomnia, and Down syndrome. And folks, this concludes the first chapter of Pathology and Microbiology for Funeral Science students by Mullins. Once again, like, share, subscribe. Let's break the algorithm. Throw something in the comments below about where you're from or what mortuary school you go to or how I'm doing. And this is Professor Finn signing out, and we will see you next video.